Okay, to everybody who's just arrived for this Zoom seminar, thank you very much for coming tonight. We have a very interesting presentation tonight, and we have a very interesting audience tonight, I see as well. We've got people from Thailand and several Asian countries, and people from Africa, South America and Australia. So it's great that we can have such an international audience. And we're very pleased today. We also have this very interesting speaker. So this is Dr. Christelle Langley, is a cognitive neuroscientist. She has a PhD from the University of Bristol, and she's been working in the last few years at the University of Cambridge, but she also does collaborations with University College London and University of Copenhagen. And she's been doing some very interesting work on serotonin, in relation to cognition and to reward processing. And in fact, some of her research has recently been getting a lot of media attention. And I'm sure she'll explain why this has been so interesting to everybody. So I will hand over to Christelle.
Perfect. Thanks very much, Graham. So I'm just going to share my screen. All right, is that sharing? Can everyone see my screen?
Yeah, it's all good.
Okay, perfect, great. So of course, I'd like to just start by thanking the organisers for arranging the seminar, and specifically to you, Graham Pluck, for inviting me to speak. So as Graham mentioned, I'm Dr. Christelle Langley. I'm a postdoctoral research associate in Professor Barbara Sahakian's lab at the Department of Psychiatry at the University of Cambridge. And today my talk is going to be about how serotonin affects cognition, and I'm specifically going to focus on our recent publication in Neuropsychopharmacology, where we examined the effect of chronic escitalopram administration on cognition in healthy volunteers. So in terms of an overview of the talk, I'm going to start by discussing hot and cold cognition. I'll then briefly explain the mechanism by which serotonin reuptake inhibitors work. I'll then move on to discuss some of the background literature, which includes acute and subchronic studies of serotonin in cognition, both in experimental animals and in humans. I'll then go on to detail our own study examining the chronic administration of the SSRI escitalopram on cognition in healthy volunteers. And then I'll also discuss some important considerations when interpreting and understanding the results of the serotonergic effects on cognition, as well as conclusions and future directions of our own study. So in more recent years, researchers have suggested that cognition can actually be separated into hot and cold cognition. And cold cognition refers to information processing in the absence of any emotional influence. So theoretically, cold cognition is engaged on tests where the stimuli are emotionally neutral and the outcome of the test is not motivationally relevant. And these include measures of things like inhibition, cognitive flexibility, attention, memory, and executive functions. Hot cognition, on the other hand, includes social and emotional cognition. And importantly, these tests may be variants of cold cognitive tests that were adapted to include emotionally valent stimuli or a motivational component, as would be seen, for example, in an effective go-no-go task or a reinforcement learning tasks. So just to explain this briefly, so the effective go-no task is an example which is based off of a well-known cold cognitive inhibition task, which is the typical go-no-go. But because the stimuli attended to are actually emotional, so they'll be using words such as happy or sad, this then becomes a hot cognitive test instead. So hot cognition is often associated with impulsive behaviors, whereas cold cognition is associated with more thoughtful and deliberate decision-making. For example, when someone is angry or anxious, their hot cognition may lead them to react quickly and impulsively without perhaps considering the consequences of their actions. And on the other hand, cold cognition may be used when making complex decisions, such as buying a car or choosing a career. And this is where careful analysis and reasoning are necessary. So of course, both types of cognition are essential for our daily lives. And they work together to help us make decisions that are appropriate for a given context or a given situation. And understanding the differences between hot and cold cognition can help us to become more aware of our emotions and how these influence our thoughts and behaviors. And importantly, there are a number of brain regions and neurotransmitters that are involved in these processes. For example, we know that the dorsolateral prefrontal cortex is often associated with executive control, whereas, for example, the orbital lateral prefrontal cortex is more associated with emotion processing. But obviously, these regions interact and they're not entirely separable. And there's been quite a lot of research, specifically by Alexander DeLong and his team. So one such neurotransmitter that is implicated in a number of brain functions is serotonin, or 5-HT, and it's a monoamine neurotransmitter. And specific drugs that target this transmission are called selective serotonin reuptake inhibitors. And they're actually the first line pharmacological treatments for many neuropsychiatric disorders, such as major depressive disorder, obsessive compulsive disorder, and also some of the anxiety spectrum disorders. So however, we don't understand the exact therapeutic mechanism of these drugs yet. So just as a brief idea of how they work in terms of neurotransmission. So during a typical neurotransmission, a message is passed from one neuron to another via synapse. And this is essentially the small gap between cells. The presynaptic cell releases neurotransmitters such as serotonin into this gap to aid neural transmission. And the neurotransmitters are then recognized by the receptors on the postsynaptic cell. And this is how the signal is relayed to the next neuron. So the majority of neurotransmitters in the synapse are then taken up again by monoamine transporters back into the presynaptic cell. And this process is typically known as reuptake. So SSRIs actually inhibit the reuptake of serotonin, and this allows for serotonin to both remain in the synapse for longer, and it also increases the amount of serotonin available to bind to receptors. And this may essentially mean that the postsynaptic cell is being repeatedly stimulated. So the increased availability of serotonin in the brain is believed to be responsible for the therapeutic effects of SSRIs. However, as I mentioned, the exact mechanism by which SSRIs achieve their therapeutic effects is not fully understood. And this is in part due to the fact that there's no way to currently measure serotonin levels in the human brain non-invasively. So a major interest for many cognitive scientists, including our own labs, is to understand how serotonin actually affects cognition. And many studies have examined the modulatory effect of serotonin on cognition in both experimental animals and in humans. So here's on the right on the left is Barry et al., who looked at rats and showed that impairing serotonin function by depleting global 5-HT disrupted reversal learning, whereas enhancing serotonin function by a single high dose of citalopram actually improved reversal learning. And similarly, in marmoset monkeys, Clark et al. Showed that serotonin depletions markedly impaired performance on reversal learning. And really interestingly, in a second experiment, their results showed that the reversal deficit induced by serotonin depletion was not actually due to a failure to approach previously unrewarded stimuli or even reduce proactive interference, but instead it was actually specifically due to a failure to inhibit responding to a previously rewarded stimulus. So in humans, the modulatory effects of serotonin on cognition have largely been examined through either acute dietary tryptophan depletion or through acute SSRI administration. And these studies have demonstrated that a wide range of cognitive functions are affected by serotonin modulation in healthy volunteers. So for acute dietary tryptophan depletion, it has been shown to affect both measures of hot and cold cognition. Specifically that it induces waiting impulsivity and impulsive behaviors. It impairs goal-directed behavior, and it shifts behavioral control towards habitual responding in appetitive conditions, but goal-directed in aversive conditions. So the effects of tryptophan depletion have also been seen on reinforcement behavior, reversal learning, learning and memory, affective and social cognition, and moral judgments. So I don't actually know if I have a pointer, but on the top right of the graph, we can see the Kanan et al. Publication. And here the tryptophan depletion group takes significantly more trials to reach criterion than the placebo group, meaning they're essentially making significantly more errors at reversal. And at the bottom, we have a study from Molly Crockett's group. And this interesting article actually shows that tryptophan depletion affected responses to unfairness. So essentially in this task, the participant works together with another player, although this is often a computer, to build a pot of money. And the participant is then offered a share of this money, which can be quite unfair. So for example, you can see here, there's either a 45% share, a 30% share, or only a 20% share being offered. And we can see from this graph that the depleted group reject significantly more unfair offers than the placebo group, and suggesting that they're more sensitive to unfairness. So studies examining the acute administration of SSRIs have shown impaired probabilistic learning and impaired cognitive flexibility, but increased long-term memory recall, emotion recognition, and harm aversion for moral judgments. However, there have also been some inconsistencies in the literature. For example, one study showed that response inhibition improved with SSRI administration, whereas another actually showed no effect. So on the left of the slide, we have a graph from Chamberlain et al. And this shows that participants taking citalopram were significantly impaired in reversal learning compared to both placebo and actually participants taking atomoxetine, which works on a slightly different neurotransmitter called norepinephrine. And this suggests a specific learning effect or a specific effect of serotonin on reversal learning. So another really interesting study, which actually very closely mirrored our chronic study that I'll be presenting, was Scandali et al. And they administered acute escitalopram to healthy volunteers. And their results showed impaired learning and cognitive flexibility, but improved response inhibition, whereas emotional processing was unaffected. Currently, only very few studies have examined SSRI administration subchronically, and this is typically over about 7 days. Short-term administration of antidepressants may ameliorate the negative biases and information processing that are often presented or present in mood and anxiety disorders. And this is work by Catherine Harmer's group. And also, quite interestingly, for reinforcement learning specifically, a recent study examined both the acute and short-term effects of SSRIs, and they showed that acute administration did not affect reinforcement learning, but that short-term administration resulted in increased learning from punishment with reduced learning from reward. So that is in the upper right-hand corner with Micheli et al. So the positive you can see in session 1, which is essentially the acute administration, there's no differences between the groups and also not between punishment or reward learning. Whereas at session 2, which is after 7 days of administration, you actually start to see that the SSRI group are learning better from punishments than they are from rewards. But interestingly, there is no official statistical difference between the performance between the acute and the short-term administration. And as such, the results of this study must be interpreted with caution. So in addition, studies with patients with MDD have shown that SSRIs impair learning from negative feedback while having only negligible effects on learning from positive feedback. And these findings demonstrate that understanding the modulatory role of SSRIs on various cognitive and motivational processes are actually quite difficult. A study that looked at a tryptophan-rich diet in middle-aged healthy volunteers for 19 days, so this is almost the next longest, showed that the emotional bias to negative stimuli was reduced in this tryptophan-rich, so actually by boosting tryptophan. So understanding the acute effects of SSRIs on cognitive processes in healthy volunteers and patients with MDD is extremely complex. And this may be due to differing possible pre- and post-synaptic actions, And moreover, there's some evidence that neuroplastic effects of SSRIs only emerge after more chronic administration, so 14 to 21 days. And as such, the chronic administration of SSRI may provide slightly more robust results. And importantly, chronic SSRI administration is also an experimental model that better mimics a treatment model of MDD, because as we know, they are the first-line treatments for many psychiatric disorders, and patients are often taking these drugs for prolonged periods of time, months or even years. So previously to our study, there weren't any studies that have actually examined the more chronic effects of SSRIs on such a wide range of cognitive measures. And it's really important to understand their long-term effects. So to do this, we recruited healthy volunteers who were given either placebo or the SSRI escitalopram for at least 21 days. And the reason we chose escitalopram specifically is because it suggested that it's one of the best tolerated SSRIs. So any interested participants were screened for a number of exclusion criteria, and these included any previous history of depression or neuropsychiatric disorders. So we wanted to make sure that they were completely healthy and didn't have any previous depressive episodes. Eligible participants were then also semi-randomized, and this was done by a staff member not involved in the data analysis or with the participants themselves. And the two groups were matched for age, sex, and IQ. So the semi-randomization in the study was actually really important because age, sex, and IQ may affect cognition by themselves. And therefore, by semi-randomizing, the participants were able to better identify drug effects rather than any possible effects due to IQ, for example. So all participants took medication daily. They either received an effective clinical dose of 20 milligrams of escitalopram or placebo, which were in identical capsules. And we were very careful to verify treatment compliance. So we actually inspected the capsule containers themselves when they attended the visits. We also took blood samples from participants, both at the halfway point and at the cognitive visit. And also participants completed a medication logbook daily. And these were also checked. And of course, both the participants and any of the investigators involved in data acquisition or data analysis were blinded to the intervention type. And so after a minimum of 21 days, participants then attended a cognitive visit. And we used quite an extensive neuropsychological test battery that assessed both cold and hot cognition, including reinforcement learning. And the outcome measures from these tests were categorized into specific cognitive domains a priori, and this study was pre-registered on clinicaltrials.gov. So the cognitive domains we included were learning, inhibition, executive function, reinforcement behavior, social cognition, emotion recognition, memory, attention, and decision-making. So as you can see, basically a very wide battery. And specifically, the neuropsychological tests included the probabilistic reversal learning task, the interleave stop signal go no go task, the three-dimensional extra dimensional set shifting task. We had 3 tasks from the Cambridge Neuropsychological Test Automated Battery, which included the paired associates learning, the spatial working memory, and the rapid visual information processing. And then also several tests from the newly designed hot cognitive battery named Emoticon. And this was a moral judgment task, an intensity morphing task, an emotion recognition task that only included the eyes, the ultimatum game, and the Cambridge gambling task. And then we also used a sequential model-based model-free task as well. So for the purposes of the talk, I'm only going to be going into more detail for 2 of these tasks, which are namely the probabilistic reversal learning task and the sequential model-based model-free task, as these were the tasks of reinforcement learning. So for the probabilistic reversal learning task, participants were shown 2 stimuli that are composed of 4 red and 4 green lines on a laptop screen. They're instructed to choose either stimulus on every trial, and the task consists of 80 trials in total, but these are split into an acquisition and reversal phases, each consisting of 40 trials. So during the acquisition phase, participants are required to discriminate between the optimal and the non-optimal stimulus. And this is essentially that the optimal stimulus is programmed to provide positive feedback, so correct, on 80% of the trials, and negative feedback, so incorrect, on 20% of the trials. And the non-optimal stimulus is programmed to provide the reverse, so negative feedback on 80% of trials and positive feedback on 20% of trials. The stimulus chosen by the participant on their first trial is always assigned as the optimal stimulus for the acquisition phase. And then subsequently during the reversal phase, the positive and negative feedback ratios associated with each stimulus is reversed. So in other words, the stimulus that was previously optimal becomes non-optimal and vice versa. So basically, in very simple terms, even if you choose the correct stimulus, you are rewarded with positive feedback saying that you're correct 4 out of 5 times, but actually 1 out of 5 times you're misleadingly told that you're incorrect. And previous studies have shown that patients with depression are overly sensitive to negative feedback, and then they respond inappropriately to the task, often by switching too soon. So the sequential model-based model-free task is quite similar, except that it has a 2-stage choice. So participants are required to collect space treasure. And to do this, they first choose between 2 spaceships. So this is the first stage choice. And each spaceship travels more frequently to one planet than the other. So for example, the blue spaceship has a 70% probability of leading to the red planet, and this is known as a common transition, and 30% probability of leading to a purple planet, which is known as the rare transition. And then the green spaceship had the opposite probabilities. So 70% chance of reaching the purple planet and only 30% of reaching the red planet. On each planet then as well, participants chose between 2 alien creature stimuli. So this was the second stage choice. And they're then rewarded with a picture of space treasure or with nothing, according to a probability drifting between 20 and 80% for each alien, with the other alien having the converse probability. So actually, importantly, with this task, the reward probability shift throughout the task, and this basically encourages participants to keep exploring their environment and different choices throughout the task to keep maximizing rewards. And importantly, all participants completed a tutorial that explained all the cover story as well as the key concepts and all the instructions via interactive example trials before actually completing the task. And this was basically just to make sure that everyone understood what they were doing and that all participants received exactly the same information. So to investigate the processes underlying learning and decision-making on the probabilistic reversal learning task, we applied a family of reinforcement learning models, and these were fitted to the data. And a similar hierarchical Bayesian modeling procedure has been used in previous studies examining this task. So for our study, the winning model actually included 4 parameters, namely the reward learning rate, the punishment learning rate, reinforcement sensitivity, and stimulus stickiness. And as we can see from the graph, there were no differences between the 2 groups for either learning rate or stimulus stickiness, but the escitalopram group had a significantly lower reinforcement sensitivity compared to the placebo group. So the reinforcement sensitivity parameter, as we've modeled here, essentially governs the degree to which a participant is driven by their reinforcement history. So it relates to how a participant responds depending on the feedback they've received on the previous trial. So our results are of considerable relevance when we consider the patient's experience of taking SSRIs chronically. So patients often report experiencing a blunting effect. And essentially emotional blunting is when patients report feeling emotionally numb and they're not really reacting to either positive or negative events. And this blunting effect has also been demonstrated for rewarding and punishing stimuli. So it's not only just related to emotional stimuli. So in light of our own results, it's also possible that the clinical effectiveness of SSRIs for depression, for example, is due to this reduced negative affect. However, if indeed positive affect is also reduced, then this would lead to a more general blunting effect, as is often reported by patients taking SSRIs chronically. And this is supported by the present study, in which lower reinforcement sensitivity would suggest decreased control over our behavior by both rewarding and punishing stimuli. And this finding is further supported by the results from the model-based model-free task. And this task is essentially analyzed using a general linear mixed effects regression, where the first stage choice, so whether they stay or switch from a previous trial, is modeled independently by whether they received a reward or not on the previous trial, as well as whether there was a rare or common transition. And the results suggest that both groups are performing in a model-based way, which indicates that they're both goal-orientated. But when specifically looking at the response to reward, the placebo group are more likely to stay, so they'll respond with the same stimulus following rewarding feedback than the escitalopram group. And again, this suggests that the escitalopram group are not as sensitive to the rewarding feedback as the placebo group. So our results showed no effects on any of the other measures of hot cognition. And interestingly, studies previously have shown that both acute and subchronic SSRI interventions do affect emotion recognition, although this is specifically for the recognition of fear and happiness. And in our chronic study, we didn't actually examine each emotion individually, but we rather examined the affective bias, and this was not affected significantly by escitalopram. And we also didn't find any effects of escitalopram on moral judgments as previously reported following acute treatment. Similarly, our results showed no significant effects on any of the measures of cold cognition. And again, this is in contrast with some of even our own previous data obtained during acute administration. So previous studies manipulating serotonin acutely, as mentioned before, have shown alterations to both hot and cold cognition. And it's important to note that obviously our study examined the administration over a much longer period of time. And this hasn't been studied previously in the context of human cognition. So in direct contrast with the present findings were those of Scandali and colleagues, who, as I mentioned, used a very similar neuropsychological test battery and showed that participants administered escitalopram acutely made more errors during stage 1 of the probabilistic reversal task, and they also exhibited increased loose shifting after misleading feedback. However, importantly, they didn't conduct the same hierarchical Bayesian modeling as we employed in the present study. And such an analysis of the Scandali data is actually currently in submission, and it does show a similar reduced reinforcement sensitivity in the escitalopram group compared to placebo controls. And we actually think that the null findings are quite encouraging.